I'm excited to introduce Sandy Kawano today. So I don't know if folks, not everyone in the room maybe is familiar with Mountain Lake Biological Station, but this is a fantastic and long-standing and very important research station affiliated with UVA in the mountains. And it is beautiful and produces so much amazing research. And she is the associate director of this new station. And we are excited to have her now out here in Virginia, also as assistant professor of biology at UVA as well. And she's going to be sharing some cool research and maybe some plugs for the station at the end as well. So thank you, Sandy.
Thank you, everyone. It's such a pleasure to be here. I'm very grateful for the invitation to speak. This is my first VHS meeting, and it's been super fun so far. So I'm generally interested in understanding, sorry, I'm short. I'm generally interested in understanding why there are so many different species, why they look so different, why they look the way they do, and how that can help them move through their environments. But I have to warn you, I apologize in advance. I started off as a fish person, still kind of like fishes too. But this was the triad, the picture that really kind of blew my mind as an undergraduate. It's called Paul Webb's triad, and it really helped to visualize a lot of what I was thinking. I started off with a lot of art, and I just was drawing all these different morphologies of birds and whatever, and I don't know why they look so different. And so this triad was really like captivating for me because you can look at the corners and see, oh, if you're a torpedo shaped like a tuna, you're really good for cruising, like constant moving at fast speed. But if you're a little bit more, you can be long and slender, but if all your power is towards the back, you might be more of an ambush predator like a pickerel or like a pike. And if you kind of have this low aspect ratio, kind of like a pancake, oftentimes you're really good at maneuvering in complex environments. So I started using this as sort of a basis to explore why are there so many animals? But I take that from the perspective of combining engineering and biology, which is a field called biomechanics. So I'm really interested in applying engineering and physics to understand the underlying machinery that allows animals to do what they do. So I combine museum collections, field-based studies, animal experiments. So I know you might laugh, but we do use high-speed video on salamanders. They can be surprisingly fast. I also use a lot of imaging techniques so I can look at some of the bones and the other anatomical structures in more detail. And then I can put that all together to create a computational model of the animal. Because as they say, if you really want to know how something works, you got to break it apart and put it back together. And so when we do that, we can actually start testing questions that you can't normally do in experiments. So as much as my salamanders are, wonderful little creatures, I can't tell them, hey, I need you to walk at a faster speed. They're going to do what they want to do. Or, hey, can you walk a little bit more like that other salamander? No, but you can do so with a computer environment. But computationally, that can be really difficult if you want to know what they're doing in very different types of physical environments. So if you're dealing with anything that's kind of fluid, like a substrate like sand, we've been using or collaborating with physicists and engineers to make robo-physical models. And so what we can do is 3D print them and be able to test questions that are really computationally intensive to actually program. And this work could not have been done with the wealth of colleagues and funding resources that have supported this research. So I was formerly at the George Washington University, where I was collaborating, or I currently still collaborate with Dr. Alex Pyron, who really kind of, pushed me into the field of herpetology and brought me back to field biology, actually. I was primarily lab-based. My wonderful undergraduate students, some of who have already graduated, including my PhD student or former PhD student, Dr. Jonathan Huey, who's now a postdoc at UC Irvine, my former postdoc, Dr. Emily Naylor, who's now at James Madison University, and Highlands Biological Station, especially Jim Costa, Jason Love, and Mike Mannion, who have been really welcoming me into the southeastern area. And I don't have to probably tell you or convince you, but we are so lucky to be here. We are the treasure trove for salamanders across the globe, and especially in the Southern Appalachians, it's just an amazing amount of diversity. If we look just within one of them, the group called the dusky salamanders, there's some really interesting stuff going on there. you know, they look drab, maybe they're not that charismatic, but they are so interesting from a genetic level. So what my collaborators have been doing is looking at the genomic data across geographic regions, different species, and just recently, so in 2021, we had 22 described species. Based off of their phylogenomic data, so a lot of molecular data, they're finding that there's now at least 47 candidate species, and some of which actually have become their own species. And it gets very complicated because once they split into different groups, they decide, hey, let's come back together again. So there's a lot of mixing going on. So if you're a molecular biologist trying to trace their genetic history, it's pretty rough. If we look specifically at one of the species, so the northern dusky salamander, it was originally, it still exists as Desmonaceus fuscus. What we can see is that within there, across its geographic range, based off all the colors you see on the left, it is breaking down into so many different lineages to the point that some of them have become different species. So the one on the kind of middle top, that is the wolf dusky. Now you can see within there, the circles are different colors. So even within that species, there's still some geographic variation within that species. But if you look down, we have the Piedmont dusky, which also broke off from the northern dusky salamander. It's actually pretty consistent across its range. So why is it that we're seeing so much cryptic speciation, so much splitting off into new species and genetic variation within a species in some groups, but not in others? And geographic variation so far is not the primary reason of why this happens. It's not a consistent answer. So that's where I kind of came into the project as more of a physiologist and biomechanist. I wanted to know why. What is the mechanism? And one of the common questions is, okay, well, if they are maintaining gene flow, they're interbreeding, what are factors that would prevent them? And so my question is, are some just better athletes than others? So the joke is that we're running the Salamander Olympics in my lab. We're starting off with 3 main categories, but we're expanding from there. So how strong are your bones? Because if you don't have strong bones, you're probably not going to be able to support any long distance dispersal. And long distance for some of them is maybe like a meter or something. Metabolism, so how much energy does it take for you to survive? Just baseline data. And then also, how much energy do you have to expend to not only do the dispersal, but then recover from it as well? And so we also incorporate endurance trials to actually see, can some of them have a capability to just move longer distances than others, or some just, they cannot physically move. It's not a matter of preference, they cannot move very far. So with all of these categories, we would expect higher values in the groups that are considered better athletes. And I'm just going to show you some sneak peeks of some of the information that we have. So first, we're going to start off with the bone strength. So just to kind of orient you as to what you're looking at, on the left side of the screen, we study their upper arm bone, their humerus. And then on the right side of the screen, we're looking at their thigh bone or their femur. These are 2 really important bones for allowing you to be able to walk. And underneath both of those charts, you can see that there's a gradient from more aquatic on the left in blue to more terrestrial in red on the right. And we're kind of seeing this consistent pattern that if you're a more terrestrial species, you have stronger limb bones. So most of you might be saying, oh my gosh, I can't believe you wasted so much time to tell us something that's pretty obvious. But here's the cool thing. It depends on not only their habitat, but also their developmental strategy. So if you look at the left side of the screen, that really dark red box plot, it's higher than that orange one. Both of them are terrestrial, but the dark red one, those are the direct developers. They don't actually have an aquatic phase. And so we're seeing some of that nuance of, it's not necessarily where you're found, but also what was the developmental strategy for you to get where you are. And there's some really cool stuff about it. I won't say, too much because I won't have time, but when you actually look at other aspects of their bone materials, the semi-aquatic ones are super strong. They're actually more strong than the terrestrial ones. And if you're more aquatic, you're actually really squishy. You don't actually break their bones. Now, you might be wondering, who is that one little blue dot all by itself there? That is one of my favorite animals, the Spanish ribbed newt. If you're not familiar with them, they're the ones that shoot ribs out of their skin as a defense mechanism. Very cool creatures. They're also multiphasic, so more on that later. So what about their metabolism? Do we end up seeing that there's this pattern where maybe more terrestrial species, they have higher metabolic rates? Not necessarily. So if you look across most of these groups here, so I have the mole salamanders, that includes the tiger salamanders. I've got those dusky salamanders. I have eastern spotted newts, and then those woodland salamanders like the Plephodon group. I mean, they're fairly similar across the board in terms of how much energy they need just to survive. And then you've got this climbing salamander group that's kind of all over the place. Well, the very top, because you can see there's a lot of range or variation to that, the very tippy top of that is actually the green salamander from West Virginia. Well, at least we collected them from West Virginia. So they're climbers, but they're rock climbers. The arboreal ones that we collected from California, they're in the middle. So the rock climbers are better than the tree climbers. And very, very bottom, even though they're called climbing salamanders, not all of them climb. So we have one group, one species from New Mexico. It is a ground dwelling Aeneades. It stays on the ground, it does not climb. And so it's kind of having a metabolic rate that's a little bit more similar to some of the other groups. So it's not necessarily just where you live, how you got there, but what are you doing to get around in your environment? So the next step was to see, okay, well, do they have better endurance? Are we seeing generally our terrestrial groups more have better endurance? And prior work by Rob Denton in Ohio was finding that if you put a salamander on a treadmill, it does actually positively correlate with the dispersal distance. So that's what we did. So this is a green salamander on our treadmill, and we picked a speed that is fairly similar to kind of their preferred walking speed. I think it was around 0.5 body lengths per second. And we just saw how long they would walk. And some of these guys would be walking for 30 minutes, and then we would get tired. And so the results came out is that there's actually a tie. We have our green salamander on kind of that second to the left. And then we have our slimy salamander over on the right. They're both terrestrial. They're both terrestrial direct developers. The green salamander is a rock climber, but interestingly, the slimy salamander, it can actually climb. It just doesn't do it as well as some of the other salamanders. And so it does seem to look like some of these patterns that are coming out from our data are starting to show that we have some competing contenders. So drum roll, please, in terms of our winners, we have got that, not surprisingly, all of our most athletic species that we've tested to date are direct terrestrial developers. And the green salamander is winning by far, followed by the slimy salamander, and then the southern great cheek salamander that we've been studying in North Carolina. A lot more going on within there, but just giving you a glimpse as to what we're looking at. And just really quickly, I just wanted to give a sense of some of the new projects that are coming out, especially in terms of my position at Mountain Lake. I'm really interested in biocollections, especially digital collections, and making that more available to the public, to education. And so I've been teaming up with Digital Life, and the goal is to create a digital zoo to preserve wildlife. And so we can do this on live animals in the field, take a lot of photos of them, and then we create 3D models of them. So I've been teaming up with the National Zoo with Matt Evans, as well as National Museum of Natural History with Kevin DeKiros. I'm sorry, Duncan. That's supposed to be Duncan instead of doubling Kevin. He is the director of Digital Life as well as an animator from Digital Life. And so we were using live animals from the zoo. This is Hero, a 40-pound Japanese giant salamander, as well as museum specimens. And we basically reverse engineered a giant salamander. So we created this 3D mesh using our photos. We gave it a skeleton. We're going to be micro CT scanning the museum specimens so we can actually put the real bones in. And then you can create this walking salamander. And one of the reasons is that when we go into physical collections, we get the color, we get the genetic data, we get the morphology, but we don't get the behavior. And that's kind of the idea, is that we can be able to preserve them in 4D. And I just wanted to end with just a little plug about Mountain Lake Biological Station. We're down near Blacksburg. We've got amazing resources. So this is one of our beautiful, it's man-made, but it's full of wildlife there. It's really, really cool. It's fishless. And within there, just around the rim, you can be finding lots of different types of snakes there, lots of different types of frogs. We've got peepers, we've got bullfrogs, green frogs, and of course, we've got our salamanders. And some really interesting stuff. I mean, you could be looking at breeding behaviors in these animals, more redbacks than you ever want to see. One of the really kind of iconic things is, or really special things, is that the type specimen, or the individuals that helped describe the Kanawha blackbelly salamander, was collected at Mountain Lake. So if you're interested, please talk to me. We always are interested in welcoming visitors to the station, either for research or for education. Thank you.
I'm curious about on your bone strength graph. You can clearly see like the average is shifting upward. But one of the other things I noticed, and this is just something I've kind of focused on a lot, is the increase. Looks like almost an increase in variability.
Yeah.
Too. And do you think there's something to that, like
yes.
some shift in the selection environment or something going on?
That is actually like spot on. The variation is really, really important. And this is just some of the data we're starting to find. This is just based off the internal morphology. There's bigger constraints when you're looking at the surface morphology for terrestrial animals, because physically it's more difficult to support a lot of the body weight. But this is just looking at one aspect of it. So they are being potentially constrained in different parts of their bone. So that gets to be really interesting. Yeah. Yeah, go ahead. Yeah, that's an excellent question. So for students who are not at UVA, are there ways for you to get involved? Yes, we have not only a research experience for undergraduates program every summer, where you get to spend 10 weeks up in the mountains conducting research with faculty members. You can also be reaching out to some of the visiting researchers, just seeing if they have any positions available. So I'm looking for students who'd be interested in working on salamanders, either at UVA or Mountain Lake. Other researchers as well. We have people coming from Swarthmore and bringing their undergraduate students as well. So if you're interested, please come and talk to us. We also have summer undergraduate field courses, including field herpetology taught by Christian Cox from Florida. So there's lots of different ways to get involved. And we also have financial aid. So don't be bashful. Please ask. We're happy to, help you out whatever way we can.
We do have a question in the chat, if there's none more in the room. The individual in chat is asking, did you have to kill the animals and extract their bones to gather this data?
Yeah, that's a really excellent question. So for the bones that I'm showing you right here, this was actually a COVID project. So my PhD student, he started right when the pandemic hit, and so he had to pivot. So these were either from museum collections, or he had to use digital repositories like MorphoBank. So that's where sharing becomes really important, because a lot of this research could not have been done without people graciously sharing their materials. But I will say that if you are actually going to be physically breaking the bones, you do have to put down you have to euthanize them in a humane manner, but it is highly regulated by the university as well as the different state scientific collecting bodies. But each specimen, everything gets used. So bones, muscles, brains are being sent out for transcriptomics. We have the genomics from the different parts of the tissues. So everything gets used, because the idea is that we're trying to create these extended specimens where we have loads of data on a single specimen to make it, the most bang for its buck, because we really want to minimize the number of animals that we're using. We want to be able to see them out in nature as best possible.